Hi everyone, and welcome back to Biology 206. This week we're going to be talking about populations, and specifically population distribution and abundance. I'm Dr. Jennifer Baltzer, and let's learn about populations. The first part of this week's lectures will focus on estimating population sizes. And so we'll learn some terminology about population size, about populations and population sizes, and we'll learn about some methods used to estimate population sizes. So first, populations. This is a definition we've already learned, but just as a reminder, a group of individuals of the same species that live in the same area at the same time and interact with one another. Okay, so lots of pieces in that. One species, all living in the same area at the same time, and they're interacting with one another. So what does interaction mean? Well, for a sexually reproducing species, this may mean having individuals reproducing with one another, so exchanging genes. But interactions can mean other things as well. We know there's lots of different interactions that happen amongst organisms, including competition, cooperation, various other interactions between individuals that can mean that those individuals are interacting. So this need not be limited to individuals that can reproduce with one another. Okay, some additional terminology here. So for populations, we have a couple of really important terms. One is abundance, and this is the number of individuals. And I'm underlining this because we're going to be focusing on what an individual is. And you may think that's an easy thing to think about, but I'll show you that it's not quite so clear-cut. So abundance is the number of individuals of a species or population. So number of individuals. So sometimes a population entails the entire species. Sometimes a population is not the entire species. So we're talking about basically the population size. So the abundance is effectively the number of individuals of that species in that population. The distribution is the geographic area where individuals of the species are present. So this is anywhere where that species has an abundance of more than 0. And we can look at the distributions of different species in relation to one another. Again, the distribution being anywhere where that species is found, okay? And here we have a couple of different desert taxa. One being the saguaro cactus, which is depicted in the brown distribution. And the other is the creosote bush, which is depicted in the reddish distribution. And so when we go out and we census where individuals occur from a species and their abundance, then we can it gives us a lot of information about drivers of the species distribution. Some of the things we've talked about in previous classes where we're thinking about environmental constraints on where a species can occur. And we'll go back to this figure in the second part of this week's lectures. It can also give us information about where species from different, or where individuals from different species may be interacting as well, of course. Yeah, but basically when we're thinking about distributions, this is the kind of thing we're thinking about, just looking at the spatial extent where that species actually is able to occur, where it has an abundance of greater than 1. All right, so back to this point about individuals. So what is an individual? And you may think in your head that you have that very clear. You know, I'm an individual, you're an individual. But it gets a little murkier when we're talking about some taxa that do have other types of life histories than we do. So just as an example, this lovely grove of aspen. You may look at that grove and say, okay, well, yeah, you know, there's a lot of different individual trees. And you're not wrong, there are, But a really cool thing about aspen is that it's able to reproduce asexually. It's also able to sexually reproduce, but it's able to reproduce asexually. And so on its roots are all of these buds that are really active. And so the buds can produce new individuals, new suckers that come up and form new stems. And so sometimes with aspen, you'll have a grove where every single individual in the population, in that grove, is genetically identical. They're clones of one another, okay? And so then you start to think about, well, what is an individual? These may all be connected below ground by rooting systems. So aspen send out these incredibly long roots. When we're studying boreal forests after fire and there's an aspen, a dead aspen individual in the stand that's been burned, we'll often find juveniles, sucker juveniles, coming off the roots of that fire-killed individual 10, sometimes 15 meters away from that individual. So the rooting systems of this species are really tremendous and have a lot of asexual or vegetative reproductive potential. Okay, so as you can see, it becomes a little bit more complicated than just being able to discern an individual when you've got all of these really interesting different life histories that different species are able to take advantage of. Okay, so, but it's really important for us to be able to identify what an individual is because population size estimates rely on measuring the number of individuals. And so, yeah, we might have thought this was easy, but then it becomes a little bit more complicated. So when we think about this, do we think about this as one tree or many trees? And in some ways it really depends on what you are interested in. Me, as a forest ecologist, as someone interested in these sorts of stand dynamics, I would probably consider these all individuals, even if they are genetically identical. But if you're interested in other aspects of population dynamics, different aspects of the reproductive biology or different genetic components, evolutionary components of within populations, then you might very well consider this to be a single individual because all of those stems are genetic. Well, they're all clones. They're all genetically identical individuals. And so sometimes when we're thinking about these complicated organisms, the way we define an individual depends on our question. Okay, so then defining the individual. All right, so this is a nice figure taken from your text, and it shows all kinds of different ways that individuals can, or that cloning happens in both plants and animals. So we have this process of budding, and so this is just building on the previous slide. We have this process of budding where a new individual is produced at some bud or node in the original individual and eventually detaches. And so we have here this duckweed where the daughter plant detaches, or you can think of a lot of succulents have those little juveniles that are produced right at the edge of the leaves and just can drop off and form new individuals. We see the same thing here in this hydra, where the main individual produces a bud that then detaches and floats away and forms a new separate individual elsewhere. But genetically, they're identical. Apomixis. This is where embryos can be produced without sexual reproduction, and so in many cases, what happens is that the egg actually is able to form directly into an embryo. It does not require a contribution from a male in the population. And so we see this in lots of different plants, to a lesser extent in animals, but it certainly does happen there. And then finally, this aspect of horizontal spread, and this is really common in plants where they send out these horizontal, and we saw this in the aspen, these horizontal either above ground or below ground stems that then produce at some point along the horizontal stem, produce roots, produce above ground biomass, and a new individual is formed, again, that's genetically identical. There are some animals that do the same thing, and we see this here in this hydroid. Okay, so all kinds of different amazing ways that plants and animals have come up with cloning themselves and producing individuals that are genetically identical to themselves. But it does again muddy the waters a little in terms of our definition of individual. And so biologists have of course come up with ways to define this. And so first of all, a genet is the product of a single fertilization event. So a single genetic individual. So whether we're thinking of the aspens on the previous slide, or whether we're thinking of any of these modes of asexual reproduction, all of the individuals that are produced via these modes are referred to as genets. So they're a single genet. So that entire aspen clone is a single genet. All of the daughter plants from that duckweed, they're all a genet because they're genetically identical. So the interesting thing though is, as we talked about in the previous slide, is that genets can be physiologically independent, compete with one another for resources, and interact with one another just the way non-genetically identical individuals would. So then, functionally, these are individuals, and so this is where it also becomes a little bit trickier. So in all of these cases, you end up with functionally independent individuals that are genetically identical to the parent plant. And so we refer to that as a ramete, an actually or potentially independent member of a genet. Okay? So the genet is all of the genetically identical individuals. The ramete refers to those functionally independent members of that genet that can interact with one another and in fact, compete for resources, etc. Okay, so again, depending on your biologic question, depending on what you're looking at, you may use this as your definition of individual, or you may use this as your definition of individual. Again, as an ecologist, I often use this as my definition, but there are lots of instances where the genet is going to be your topic of interest. Okay, yeah, so the take-home message is that the definition of an individual is really question dependent, and you really need to think about, for your particular question, what is the relevant definition for an individual for me, and what does that mean for my sample design? Because again, if you're looking at, for example, if you're looking at the response of a species to some treatment, and you only have a single genet because you've happened along, an aspen grove, for example, that's a single genet, well, the response you're getting is not going to necessarily be indicative of what that whole species, how that whole species would respond, because you have a single genotype that you're actually testing. And so it becomes very tricky. And so it's really important to consider which of these is the appropriate definition. Okay, so we know what individuals are, we know that it's complicated, we know that you really have to carefully think about your question in order to identify which of those definitions is the right definition for you. But what do we do now? Well, we count. This was one of my kids' favorite songs, Do the Batty Bat, and the count goes through and counts all of the bats and laughs maniacally. Anyway, so I had to include that for you. But we count, okay? So in theory, that's pretty easy, but it turns out that that's kind of another no. Just imagine being in this beautiful field of wildflowers, and it's your job to measure the population densities of those species occurring in that field. Just going out there and counting isn't going to work very well because there's just such a high density of individuals. You're going to lose count of where you are. You are not actually, and the numbers are too high. You would never be able to complete that census of that whole field, and that's just a small patch of that field. Okay, so we have to come up with ways to estimate abundance, to estimate population densities without actually counting everything for most situations. You know, in some species where there's very few individuals, for example, you probably are going to want the exhaustive count. For many species, what you're going to do is you're going to use sampling methods that allow you to estimate the population size. Okay, so how do we do that? Well, again, we need to know what we need to know. So there's a couple of types of population sizes. First is the absolute population size, and that's the actual number of individuals. So for some studies, it's really important to get as close to that actual number of individuals as you can. For some studies, a relative population size is okay. So that's the number of individuals in one time interval or place relative to the number in another. And so a lot of times what we use is measures that correlate with the absolute population size. So this could be, you know, here we have a beautiful lynx, a beautiful Canada lynx. You know, if you were estimating absolute population size, you might put colors on those and do some kind of tracking of individuals, some kind of mark recapture, which we'll learn about. You might set up some kind of camera traps where you're able to identify features of individuals, or you know, there's different ways that you could actually directly estimate the absolute population size for an area or the actual number of individuals. But the relative population size, there are various ways you might do that. You might use tracks. So you might do transects in various places and count how many times you come across tracks. You might count how many times you come across scat. So yeah, it can be tracks in an area. If you're interested in fish, maybe that's fish catch per unit effort. So, you know, how long your trawler was down there for, how many hours you spent trying to catch fish, whatever it is. So we think about that catch per unit effort. It's a relative population size, comparing it to some other time or some other place. Bird songs or sightings along a fixed distance. So you're not going to get the true population size, but if if you're hearing, you know, you're hearing robins singing, you know, 15 times along your transect in one place and only 5 times along your transect in another, that your relative population sizes differ, okay? So, absolute population size, relative population size. And there's a range of methods for sampling both of those. We're going to focus really on these absolute population size metrics, but a lot of the methods would be comparable for these kinds of correlates of absolute population size that you use to estimate relative population size. Okay, so first up is area-based counts. And this is exactly what it sounds like. You have some fixed area, you count the number of individuals in it. Sometimes we score presence absence, sometimes we count, and I'm going to show you that sometimes we also estimate percent cover, and that it really depends on what you're measuring. And you can see this scuba diver doing this on a coral reef, and this is very typical kind of infrastructure for doing that. It's pretty fancy. It's typically 4 1-meter long PVC pipes with little PVC corner attachers. That's all there is to it. And so as a forest ecologist, these are super great because you can just pull them apart, have the sticks, pop them in your bag again, and then reconstruct when you get to your next site. Okay, and so you have your quadrat. Typically, a 1x1 is a very common size, but the size of your quadrat is really going to be dictated by your organism in question. And so we have a little example here where maybe you use a 1 meter square quadrat for the herb layer, but for the shrub layer you use a 5 meter square quadrat, and for the tree layer you use a 10 meter square quadrat. And the methods you use to estimate within those are a little bit different, but basically quadrat-based sampling it's not always the same area that you sample, but you always have to take note of that area because you're counting a number of individuals within an area, and that gives you a density. And then you can do that over and over again in different places in the landscape of interest, and then scale that up to get a landscape level or a number of individuals per hectare type of measure. Okay. All right, so the basics of how you might do this, it's very common to sort of want to have a random kind of location of your quadrat, so there's no bias introduced in the placement. So maybe you're interested in this sampling area here. You stand in the corner, you turn your back, and you throw your quadrat over your shoulder, and wherever it lands, that's where you sample. And there can be a little bit more strategy in quadrat sampling and any kind of sampling design. You may say, okay, I have these 5 habitats that I want to sample within, and then you subsample using this kind of method within each of those. That's referred to as a stratified random sampling design. This is just a random sampling design where you have some sampling area, you just toss your quadrat randomly within it, and you measure those places. And that sort of unbiased way of sampling is really, really important. But again, sometimes you'll stratify across habitats so you can get estimates within each of those. Okay, so you throw your quadrat, it lands, and then you figure out, well, who am I going to count within this quadrat? And so the basic rules are anything for plants, anything that is rooted within that quadrat gets counted. And so here we have these little red flowers, 3 of them are rooted within there. One of them looks like it probably isn't, so we don't count that one. And we don't count any of these that are outside of the quadrat. So it's pretty simple when it comes to plants. You just have to figure out where that plant is rooted, and you don't count it if it's outside the quadrat. You do count it if it's inside the quadrat. And area-based counts like this are typically done on immobile organisms, so plants or animals like coral that are fixed in place, or sometimes for really slow-moving organisms. So you could use this kind of method, for example, for snail population or slug population densities. You know, organisms that really don't move very quickly, and that you can count the whole population there in that quadrat. So long as, with those mobile organisms, so long as you're not interested in monitoring that precise location. So frequently with plants, we'll mark the quadrat locations and return to those same places and measure the same place through time, and then you can actually kind of get a measure of population dynamics within that quadrat. With mobile organisms, even if they move slowly, you obviously can't do that, but you can use quadrat-based methods on slow-moving organisms. Okay. So yeah, your size of quadrat is determined based on the size of the organism in question. You have a known area of plot sizes, and that allows an estimate of the density within a larger area. And this is really important. The more quadrats you sample, the more accurate your estimate. If you only put 1 quadrat down, and that you can imagine that field of wildflowers in our previous, a couple of slides back, if you put 1 quadrat down there, you are not going to get a full measure of diversity in that field. You're not going to count all of the species that are there. And B, you may end up in a patch where there are a ton of daisies or a ton of poppies, but that's not representative of the whole field. And so the more quadrats you put down, the closer you get to the true answer of the estimate. So usually as you know, as a field ecologist, a lot of times what we're thinking about is the trade-off between, okay, we want to have as many quadrats as possible, but we only have so much time at any given site, and so that's where you're trying to find that that balance between those, or the sweet spot between those two competing factors. Okay, so I mentioned sometimes you'll want to measure percent cover, and so a lot of times when we're doing percent cover, not just presence absence or not just counts, we'll do this kind of thing where you either string up the quadrat as you see done here. Again, we're underwater in this one, or a lot of times, again, because it's really hard to move through the forest with a quadrat that's all set up like this, we'll often, in my group, tape off 10 centimeter sections on these 1 meter quadrat arms and do it that way, okay? But what you're trying to get at when you're doing percent cover, it's where you have very abundant species that count it, or either very abundant species where counting them is going to be really hard, or species where counting them doesn't really make sense. And a lot of times we're thinking about ground cover, or in this case, the cover of the ocean bottom. And so then you're thinking about not, you know, and very, very often we use that for species that it's really hard to distinguish individuals from one another as well. So doing individual counts doesn't always make sense. So percent cover is very often used in plant ecology. and I imagine also in coral reef biology. And so what you do in that context is you get to your plot, you identify all the species that you have in there, and then you try to figure out, well, how many of these little subgrids do they occupy? So if we look at this white kind of brainy looking thing down here, we'd first identify how many of these squares is it completely filling, and we see it looks like maybe 3 squares are completely full, and we know each of those, if this is a meter long, this PVC pipe, we know each of these is 10 centimeters by 10 centimeters, okay? So, or 1% of 1% of the grid, okay? And so we might look at this and say, okay, we've got 1, 2, 3 that are complete, and then maybe you can I often try to sort of imagine shifting some of this one into this one. So then maybe a little bit of this combines with this to make 4, maybe these 2 combine to make 5. And so it's a little bit, you can see where I'm going with this. You know, you try to add up these squares, and if I were to look at this, I'd say probably 1, 2, 3, 4, 5, 6, maybe 6 or 7% cover of this particular species, and that's assuming it doesn't occur elsewhere. It may occur up there. But you can see the take-home message from this is that those percent cover estimates are pretty subjective, and this helps reduce the subjectivity, having these grid cells. But it's really important when you're using these kinds of methods to train everybody on the team so that you're calibrated against one another. And it's really hard to compare percent cover estimates from one study to another when you have different observers. So some challenges with percent cover, it's also very time consuming. So it's really important information if you need it, but in some cases it's not necessary to record that level of detail. A more quantitative way to get at those percent cover estimates, but again, much more time consuming, is pin frame methods, where you actually either have your quadrat set up like this, and you drop pins down at every intersection point and record which species the pin actually hits. So you can imagine doing this along one of those rows where you drop down and you hit this, whatever this grassy thing is here. There's a miss here, it hits this one, you can see you're going along and doing that, or in the same picture down here where you're hitting this green leafy species repeatedly across this transect or across the row in that particular pin frame. So this gives you a much more objective way of doing that than the percent cover estimate does. Okay, so pin frames. Again, kind of awkward to carry in the field. They take a lot of time, but they reduce that subjectivity. It's a much more objective measure than is percent cover. That said, you don't hit everything because you only measure in certain locations. You may miss species that don't fall within your pin frame in a particular spot. So there's that disadvantage to that method. Okay. But regardless, all of those things give us an area-based method of counts or presence absence or what have you. And what we can do is then scale those up to get a density. And so here we have some area that we're interested in, and we can sample quadrats. And in this case, this is showing you a few different things. This figure is showing you quadrats of different sizes and what that means for the precision of your estimate. So again, this is why it's really important when you're using these area-based estimates to try out some different sizes of quadrats and see what is the right size of quadrat for your particular taxa, okay? If you're looking at a larger organism and you're using a quadrat that's too small, you're very often going to miss that, and so we see that exemplified here in the half meter by half meter quadrat, where in 0 out of 5 of those quadrats, do we find the species of interest? But when we look at any of the larger quadrats, we find that species pretty regularly, okay? So the precision of your estimate is going to be dependent on the size of your quadrat and the frequency of your quadrat. If we used a small quadrat but did a lot more of them, that's also a trade-off, is your size number trade-off in quadrats. If we did a lot more of these small quadrats throughout this area, we might have very well gotten a better estimate of that population size. But because we only did 5, in none of those 5 small quadrats did we find our species of interest. In our 1 by 1, so the second nest, we had in 2 out of 5 of them did we find the species present. So this one and this one. So you record 2 out of 5, or 40%. And then in our 1.5 by 1.5 meter quadrat, we found individuals of that species in 4 of them. So 1, 2, 3, 4. So in that case, we see an abundance frequency of 80%. So you can see how different size quadrats lead to very, very different estimates. And so what you would, in an ideal world, if you'd sample the same area regardless of the size of your quadrat. So even if you were using half meter by half meter, you'd still measure 2.25 meters squared. So this is, when we talked about catch per unit effort a moment ago, this is the same kind of idea. Your sampling intensity or your sampling effort also determines the precision of your estimate. So your quadrat size and the total area that you sample. They're both really important pieces. Okay, but what I wanted to show you here was how then to calculate the mean density based on these counts. And so here's the equation. Your mean density is going to be the sum of the number present per quadrat divided by the number of quadrats. So in all cases, we have 5 quadrats. We have different estimates of the number. But in, let's say, the 1 by 1 meter quadrat, we had 2 instances where the species was present in the quadrat. So that's 2 divided by 5 because they're 1 meter square quadrats. So 2 individuals just divide by 5 meters squared. And then divide that by the quadrat area. In this case, our quadrat area is 1 meter squared. So we get a sampling, a mean density of 0.4 individuals per meter squared. So the sum of the number present per quadrat, you sum them all up, you divide that by the number of quadrats, you divide that by your sampling effort. So this standardizes for your sampling effort. Okay, so, and then when we look at that for the 1.5 by 1.5 meter square quadrats, although we had 80% frequency of finding the species, because it's a larger sample area, we get pretty comparable estimates of individuals per meter squared, so 0.4 and 0.5 meters individuals per meter square. Okay, so this is how you calculate mean density from those area-based estimates. So pretty simple. And then, if you sample intensively in a landscape, that actually gives you a pretty good estimate of the total population size in that landscape. Okay, now moving on to what we refer to as distance methods. So, using quadrats is not ideal in every situation. A lot of times either if you have mobile organisms, for example, or you're trying to sample across a much larger area, or you've got larger organisms. In this case, we see these trees along this transect. Maybe what you want to do instead is run a transect across your landscape and sample at very typically randomly selected points along that transect. So again, we introduce that stochastic component, that random sampling, to avoid bias in our population density sampling. Okay, so our transect starts here, it ends here. We've done 3, we've identified 3 locations on there that we're going to sample. And at each of those locations we make some measurement, and that depends on the organism in question. And one of the things that your text in the ecological, there's a box about these 3 different sampling methods that we talked about: the area-based, the distance-based, and the capture mark recapture. it talks about aspects of visibility and detection of individuals as distance from the transect gets greater. And so you can imagine if you're doing this walk and you're looking for birds, you may be much better at, well, you will be much better at seeing those individuals that are close to you than seeing those individuals that are far away. And so this is often what happens, is that we want to take a look at, well, how well are we detecting those individuals when we're walking along this transect and measuring the distance to an individual along the transect. And so in some cases, maybe you get everybody. Maybe it doesn't matter what distance you are from the transect, you have a similar number of observations regardless of the distance. And that means you don't have any detection bias. You don't have any detection or visibility challenges. But it may be the case that you're much better at seeing individuals that are quite close to the transect. And in that case, you end up with this kind of curve where you have individuals close to the transect that are well detected, and then further away you sometimes get them, but you miss them a fair amount. And that's a real problem when we're estimating population sizes. If there's a lot of false negatives, so you don't see something but it's actually there, you get a big underestimate of the population size. And so we have to take that into account. And so what we often do in that situation is we say, okay, well, I get good detection out to maybe this fourth bar in both cases, or maybe you're conservative and you say out to that third bar. And in that case, we use that narrower belt along the transect to calculate our density estimates based on our counts. And sometimes, of course, you miss things even though they're close to you. And so you need to understand how frequently, and that's what this gray hatched bar is, how frequently you're actually missing those individuals that are close to you. Maybe they're very quiet and still and you don't see them. That's a harder problem than this particular one. Okay, so again, our just an example of the detection function. And so here we have a number of detections, 5 from 0 to 10 meters, 7 from 10 to 20 meters, and then it drops right down to 3 and down to 2 and down to 1 at those 10-meter intervals. So we have this sort of situation where we're detecting them pretty well up to 20 meters, but not very well past that. Okay, and so then when we're thinking about density estimation, when we have these detection problems or these visibility problems, what we do is we build in that we say, okay, well, the width of our sampling area only goes out to that distance where I'm confident that I was catching all those organisms. Okay, so in this case, our equation is D is our estimated density, N is the number of objects observed, so we have our number of detections, so that's N, divide by the distance from the line where the detectability is assumed perfect, so that is, in this case, 20 meters, and the length, so the product of the length and the width, which gives you the area. Basically, you're doing a belt transect sampling effort, and you need to know the width of that belt and the length of that belt to calculate the area. So this term down here is our area, and it's 2WO because we sample on both sides of the transect. If you were only sampling on one side of the transect, you'd only have WO times L, but you were sampling on both sides, so we have 2 times WO, which is that width that we're confident that we're sampling and catching all those organisms. 2WO times L, so this gives you the area that you sampled. You have your number of individuals over your area. And so if our line transect for this example was 100 meters, there was a total of 18 birds detected, so we could sum this up. All of these blue bars sum to 18, but we know that we're only confident in our detections up to about 20 meters. So in that case, we had 12 individuals within that 20 meter band on each side of our transect. So 12 divided by 2 times 20, so that's the width on either side, times the length, which is 100. So we get 0.003 birds per meter square, or 30 birds per hectare. So this is, again, same idea as for our quadrat sampling. We need to know an area, we need to have a number of individuals, and this lets us extrapolate out to a density-based estimate. Another example of a distance method, and this is really commonly used in by plant biologists, by forest ecologists, is this ordered distance method. So again, you have a transect, so you're walking along a transect, you have some random point that you're sampling at, and what you do, because we're working in this case with immobile organisms, what you do is you find the nearest 3 individuals, and you measure the distance to the furthest 3rd individual. And you could do this with any number of individuals. You could say, I want to use 5, not 3. And it really depends on the density of your individuals in that area. But what you do is you measure out to the distance from your transect to that third nearest individual. And so then basically what all you do is you calculate the area of that circle. You have the radius here, so pi r squared gives you the area of the circle. You have the radius here to this individual. You know that there's 3 individuals within your circle, so it's 3 divided by whatever the area of this circle is. And you can see that in our first point it was a pretty big circle, and our second point it was a smaller circle. So the density here is going to be higher than it is here. And so we do this repeatedly along the transect at various randomly determined locations, and then we take the average of all of those density estimates that we measured along that transect, and that gives us our population density. Okay, and so our last method is, your book refers to it as mark recapture. I think of capture, mark, recapture. That reminds me of what you actually do, which is capturing a bunch of individuals, mark those captured individuals in some way, and we can see different ways that animal biologists mark these. So here we have a snail with a little thing painted on its shell, a little number 87. Here we have, not exactly sure what this animal is, but some mammal that has a collar with some unique identifier on it. These these damselflies have unique identifiers drawn on their wings, probably in Sharpie marker. Biologists do that a lot. They use Sharpies for everything. And then this bird has a bird band, that band has some unique identifying information on it. And so these are some different ways that biologists mark animals. So they catch them, they mark them, and release them. Okay, and then they recapture them. Okay, and it's that information, that known of, okay, I sampled this many individuals in this time period, I marked and I marked them all, then I released them into the real true population. So you know what your initial population of marked individuals was. You release them into the true population, they mix around, and then you take a sample again, the number of marked individuals within that sample. So some of them will be marked, some of them won't, but that's really important for getting an estimate of population size. And the way we do that is, you can imagine, so here's our definitions for all of these terms. So if we think about the ratios, this is just, kind of simple math, but the number of individuals recaptured over the number of marked individuals is kind of the marked population, right? So how many marked individuals did you catch on time 2 versus how many did you mark on time 1? And then equals the total number of captured individuals the second time. So all of the individuals you captured that second time, both marked and unmarked, divide by the actual number in the population. Okay, and then if we reconfigure that, we can get this term, which is the number of individuals in the population equals marked times total, so the number of individuals captured and marked in time 1, times the total number captured the second time with and without the mark, divide by the number of individuals recaptured, so those with the mark. Okay, so the ones that you caught the first time and the second time. And this gives us an estimate of the actual population size. So just as an example, we can think about these lovely snails here, and we would have painted on their shells and put little numbers like we saw on the previous slide. So this is just a little hypothetical situation. Your neighborhood has a population of snails. They come out onto the sidewalk when the sprinklers are on, and the rest of the time they hide in the vegetation and the grass and people's lawns, etc. So if you go out when the sprinklers are on and you capture 20 of these, you paint your unique identifier on their shells, and then release them. A week later, they've dispersed into the population. They will have intermingled with that broader population. You catch another 15, and 6 of them have marks on them. So 6 out of the 15 that you've caught have marks. So we can use that formula to estimate the size of the population. So our M is the number that you marked and that you captured the first time and marked, and so that was 20. The C is the number that you captured the second time, both marked and unmarked, which is 15. So 20 times 15, divide by 6, which is the number of recaptured individuals were marked from the first time. Okay, so then we get a population size, a total population size for that snail population of 50. So it's a pretty simple calculations, but knowing how many individuals you put out into that population that returned to you on that next time around is critical for determining population size. Not surprisingly, and you probably were expecting this, there's a lot of assumptions associated with this. So the first being, if you go out and do your recapture and you don't catch any individuals, then you can't estimate a population size. It's undefined because your R equals 0. And again, if you divide something by 0, we know that there's no solution to that, okay? All right, the marking technique must not harm the individual or affect its survival by predation. And so this is a really important assumption. If you're putting a tag onto an individual that makes it super apparent compared to conspecifics within the population, you're going to bias those individuals toward being captured by predators, for example. So we assume that there's no harm caused to the individual that affects its survival. The mark cannot wash off or wear away, and so you have to have something that's permanent. If you put a mark on something, if you draw on the shell of a turtle with chalk and then come back a week later, of course it's not going to be there, and you will have no idea which of those individuals were marked by you. So the mark has to stay on. And then there's some assumptions about population dynamics. So we know that a population size is a function of birth and deaths. So we have births, which are new individuals in the population, deaths, which are individuals leaving the population. Immigration is individuals coming in, and immigration is individuals going out, and emigration is, sorry, reverse that again. Immigration is individuals coming in, emigration is individuals leaving the population, okay? So it assumes that that populations are closed, okay, so that there's no individuals coming in or out of the population. So no immigration or emigration, it's a closed population. And it assumes that there's no mortality between the mark and recapture times. So if you mark individuals in 1 year and come back 5 years later, probably your capture, mark, recapture method's not going to work very well unless these are exceptionally long-lived and long-lived individuals that don't have a high mortality rate at all. So you have to know something about the biology of the species in order to know the frequency that you have to recapture them to get at a good estimate, okay? And the marking experience must not make an individual more or less likely to be recaptured. So we don't want to teach animals either to be afraid of being captured or to be encouraged to be captured. So if you do something that makes being captured super fun for the animal, like providing all kinds of additional food resources, for example, that may increase the likelihood that that individual is going to take advantage of being captured again. Alternatively, if you do something that really terrifies the animal, it's going to learn from that and maybe shy away from being captured. And so you have to be, there's assumptions that this capture, mark, recapture method is actually, each time you have an equal chance of capturing any individual from the population. Okay, so these are some of the assumptions. So if you end up using any of these kinds of methods in your own research, it's really important to be aware of these really important assumptions of this method. Okay, So the last couple slides, just to show you some really cool new methods that have developed for estimating population densities, estimating abundance within a population using satellite-based methods. And so these are both images taken using Worldview satellite from Digital Globe. They're images taken from skywatch.com. But in this in this image, it's each of these little black specks is an emperor penguin. And so we have a huddle of emperor penguins here. And so you can imagine being able to simply, go through and count the number of individuals in the population. So we start to be able to, with some species, with some populations, we start to be able to use the same kinds of methods that we use for area-based estimates when we're walking through the forest and putting down our quadrats or doing our transects, but being able to do this using satellite imagery and get information about a much larger proportion of the population or about much larger geographic extents that maybe you couldn't actually get these estimates if you were on the ground, for example. Really hard to reach places like Antarctica. Hard to measure individuals like these gray whales that we see 4 individuals here from the ocean, and then a boat over there. So pretty amazing things that we can do using satellite imagery to estimate population sizes. And there's also some really cool stuff happening with citizen science where images like this are put up and individuals in the interested citizens contribute to the estimates of populations. So anyway, lots of neat things you can do with this kind of satellite imagery for population density estimates. And it works for some plants too. And so here's a beautiful aerial photo of a system that I do some work in. So all of these, hopefully you recognize the biome, but this is the tundra. All of these little scaly looking things, it's polygonal terrain. So those icy wedges in the soil that create that polygonal structure in the soil like we see here. But what I'm showing you is patches of alder shrubs. And so these are nitrogen fixing, so it's Ulmus ulnabetula, and it's a species that forms a partnership with a nitrogen-fixing bacteria. It's a very tall tundra shrub that's expanding pretty rapidly in some parts of the tundra, and we can actually see these from various types of remotely sensed imagery, and we can quantify populations at fairly large scales using various image analysis. And so this is one great example. Another great example comes from the tropics, where we have really spectacular changes in the color of the canopy of certain species that allow them to be very easily picked out using spectral remote sensing techniques. So we have in yellow here one tree species, Hydranthus guaiacan, and the blue over here is Jacaranda copiae. And both of these, you have to know something about the biology to be able to know who's flowering at a given time or when to take the images. But when you have this kind of distinction between the green canopy all around it and the bright yellow flowers, so bright yellow flowers or blue flowers over here, this really allows us to use spectral information to pull out the frequency of those individuals in the canopy of the rainforest as an example. So some pretty amazing technologies coming along with remote sensing of populations and species distributions that will only continue to advance as the imagery we have available to us improves. And with that, I will leave it. We will come back to the second half of our discussion of population, which really has to do with distributions and drivers of distributions, drivers of abundance on the landscape in our second lecture this week. And I will see you on Tuesday in student hours. Have a great night.